Hello everyone, welcome to the Module 3 and Module 4 of our Molecular Biology and Diagnostics. For today's lecture, we will be discussing the DNA replication. Please do take note that this is going to be a two-part lecture video, as this is a more complex topic compared to the ones that we've discussed before. So si DNA replication, Module 3 and Module 4 po talaga ang coverage niya. And before we dive into our main topic, these are some of the few reminders. Number 1, please do advanced reading. Number 2, use your notes, module handouts, and reference books. Number 3, take down important keynotes. And lastly, sit back and relax. So DNA replication is one of the more complex topic of our molecular biology and diagnostics. So kaya po siya two-part lecture video and then 2 modules yung coverage niya. So I am highly encouraging everyone to please do advanced reading and please do take down important keynotes. Okay po ba tayo doon? And then again, let's have a quick recap or review of our DNA structure. As we all know, we are being comprised of billions of cells. And each cells, it has 3 major components. We have your cell membrane, the cytoplasm or the jelly-like fluid where your organelles are being embedded, and lastly, your nucleus, the control center of that particular cell. And as we all know, our DNA is being located at this nucleus, particularly embedded in your chromosomes. Okay? So, itong chromosome na to, meron tayong DNA dito na naka-coil, diba? Na compact siya and then coiled. And what is the protein that is responsible for the coiling of that DNA? And that is what we call our histones. Okay, yung histones mo, meron tayong lysine and arginine that makes our DNA coiled. Okay, so important po na nakakoil ang ating DNA sa ating chromosomes kasi coiled siya and then compact, mas dense para mas magkasya siya sa inyong chromosomes. And ang protein na responsible doon ay ang tinatawag nating histones. Hindi man po siya dito visible, pero histones ang tawag po doon sa protein na po iyon. And then, pagka pinag-combine na natin yung histones and then yung ating DNA, ang tawag na dito ay nucleosome or nucleosome. Okay, yung parang macaroons na parang grape-like. Kapag ka naka-coil na siya, na nag-combine na si histone and then si DNA, ang tawag na po sa kanya ay nucleosome. And then naka-compact na siya sa ating chromosome that is particularly embedded in our nucleus. So yun, quick recap lang po. And then again, what is the chemical structure of our DNA? We have 3 main groups. We have your phosphate group, your sugar group, and then your nitrogenous bases. If you will look closely on our phosphate group, tingnan po, meron po siyang negative charges. So that is the chemical functional group that is responsible for the negative charge of our DNA. And then if you will look closely sa ating pentose sugar, pentose meaning meron tayong 5 carbon sugar, 1, 2, 3, 4, 5. Yung 5, nakakonect po siya doon sa ating phosphate group. And then ang tawag sa connection between the pentose sugar and then your phosphate group, that is what we call your phosphodiester bond. PB for short. Okay po. Doon sa 5 and then sa 3. And then ang difference naman ng pentose sugar ng ating deoxyribose nucleic acid and then your ribonucleic acid is a single atom, which is your oxygen. Sa deoxyribose, wala kang oxygen. Sa ribose na RNA mo, meron kang oxygen. So that's what's make our DNA more stable compared to our RNA kasi wala siyang oxygen atom. Okay po? And then yung third major component natin ng ating mga nitrogenous bases. Okay? If you will look closely sa ating right side, we have 2 types of your nitrogenous bases. We have your pyrimidines and then your purines. Kapag sinabing pyrimidines, cyclic ring. Single cyclic ring lang siya. Okay? Pag purines, 2 cyclic rings. Kitang-kita naman po sa ating chemical structures. Sa pyrimidine, cytosine, thymine, and then uracil ang ating mga pyrimidines. Mga single cyclic ring lang. Pagka naman dalawang cyclic ring na yung pinag-uusapan, purines naman po yun. Or your adenine and then your guanine. Okay? And then, if you will look closely again and again, ang tawag po dito sa bond between the pentose sugar and then your phosphate group is what we call the phosphodiester bond. And then, ang nagko-connect naman sa ating pentose sugar and then the nitrogenous base, anong tawag dito? Glycosidic bond. So, ang glycosidic bond, applicable po siya sa between the connection or what joins your sugar to sugar or pwede namang sugar to other functional group. And then in this case, sa nitrogenous siya nakakabit. Okay, ang tawag po doon ay glycosidic bond. So what part of the DNA is found at the center of the molecule? Is it your amino acid, your sugar, your bases, or your phosphate? Okay, tignan natin sa picture dito. So kaya po naging backbone ng ating DNA. So kung mapapansin nyo, anti-parallel nga sila to one another, di ba? Yung dalawang helix natin dito. And then yung ladder natin, ito yung mga nitrogenous bases natin. Kaya po sila naging backbone, sila yung parang backbone or yung pinaka-support ng DNA, yung alternating sugar mo and then yung alternating phosphate group mo. So, that leaves our bases sa gitna. Ang sagot po dito ay bases. What part of the DNA is found at the center? Sino ba yung nasa gitna? Siyempre, yung mga nitrogenous bases or your bases. Yun po yung sagot doon. Next, weak hydrogen bonds are found between which molecules in the DNA? Is it between your purines, between your acids, between your bases, or between your sugars and phosphate? So, meron po tayong dalawang helix dito or may dalawa kang naka-anti-parallel nga ang ating mga DNA strands, di ba? So, dapat meron pa rin silang connection yung between dun sa nitrogenous bases to another nitrogenous bases. So, hindi naman pwede nakahang lang sila doon. Meron pa rin po nagko-connect sa kanila doon. At ang tinatawag natin doon ay ang hydrogen bonds. Okay, sa ating complementary base pairs, si adenine, kapartner niya si thymine. Si cytosine, kapartner niya si guanine. Pareho po yung parehong pair na yon, meron po silang hydrogen bonds. Ang pinagkaiba lang nila, dalawa yung hydrogen bonds mo kay adenine and then kay thymine. Kay cytosine and guanine, tatlo yung hydrogen bonds mo. Okay? Pareho po yan, may mga hydrogen bonds. Nagkaiba lang sa number. Okay po ba? So, to answer our question, weak hydrogen bonds are found between which molecules in the DNA? Ang sagot doon, between your bases. Kasi nga po, between your nitrogenous bases na andoon ang ating mga hydrogen bonds. Okay? So again, our DNA, meron tayong 10 base pairs for each groove. Ano po ba yung tinutukoy yung groove? Yung parang pinaka-turn. Diba? Yung intern dito. Meron tayong, sabi daw kada turn na yan, may 10 base pairs. And then, we have your major groove and then your minor groove. Ano bang significance ng major and minor groove to our DNA? The major and minor grooves are sites of interaction with the many proteins that bind to specific nucleotide sequence in DNA for the binding or recognition site. And then ito. Okay, example lang po ulit ng ating chemical structure of our DNA. Showing the phosphodiester bond, okay, yung 5-3, and then the glycosidic bond, kaya po tinawag na 5-carbon sugar, pentose sugar, and then your nitrogenous bases. So according to Chargaff's rule, diba, meron tayong complementary base pairs, the complementary strands due to base pair rulings, the two strands are complementary. So, meron kang 5 sa taas and then 3 dito. Saan ba galing yung 5 and 3? Sa 5, meron kang phosphate group doon sa dulo. And then doon sa 3, meron kang OH na functional group. Okay, so kung titingnan natin dito, 'di ba, bibilangin natin yung kanilang sa chemical structure nila, yung bilang, doon po tayo nagbe-base sa 5 and then sa 3. Kapag kasi adenine yung nasa taas, dapat si thymine yung nasa baba. And likewise, kung thymine yung nasa taas, adenine yung nasa baba. Kasi ang magka-base pair lang talaga si adenine and then si thymine, tapos si cytosine and then si guanine. Okay po, the two strands of DNA double helix are anti-parallel. Their chemical orientations are different. By convention, a base sequence is usually written with the P5' P terminal end on the left. Please do not forget. And then we have your double helical model. It suggests that the strands can, number 1, separate, and number 2, act as a template for the formation of a new complementary strand. We have 3 types for that double helical model. We have your conservative, semi-conservative model, and then your dispersive model. So first, i-discuss muna natin si conservative replication. Conservative replication is the parental DNA strands remain together in one of the daughter cell, and then the newly synthesized DNA strands went to the other daughter cell. So if you will look closely on our right side, kunwari ito si DNA na original, yung parang old template. And then kapag kagagawa na daw ng panibagong DNA, pagka magre-replicate na si DNA, according to the conservative model daw po ito ha, the parental DNA strands remain together in one of the daughter cell, and then the newly synthesized DNA strands went to the other daughter cell. Parang nandito. Okay, so siya, ganito, nag-split lang siya and then nag-split lang din yung daughter cell. Next naman, for the semi-conservative replication, each daughter cell received one parental DNA strand and one newly synthesized complementary strand. Parental strand was the template. So, ang difference naman conservative to semi-conservative, kunwari ito yung original or yung old na DNA mo. So, kung magre-replicate na yan, makakakuha siya ng old and then new. And then old and then new. Parang meron kang lumang template, tapos gagawa ulit siya ng new. Okay? Parang may mamamana talaga. Yun yung difference ni conservative to semi-conservative model. And then, meron din tayo dito sa tinatawag sa ating molecular biology and diagnostics na Mesoson style experiments where they demonstrated the semi-conservative replication, and then they made use of the bacteria and the nitrogen. So after ilang experimentations, napatunayan nila na mas applicable si semi-conservative replication sa ating DNA replication compared to the other models, yung si dispersive and then si conservative. Ito mga theories yan before. And then as time goes by, napatunayan nila na si semi-conservative model ang mas applicable sa ating DNA replication these days. Okay po? So for the overview of the DNA replication, meron tayong mga babanggitin na principles na dapat nating tandaan, dapat nating i-take down note, at dapat nating kabisaduhin. Okay po? So for the replication of the DNA, like what I've mentioned, napatunayan nga na sa semi-conservative model, ang mas applicable na theory with regards to the replication of the DNA, kung saan nandoon yung parental DNA. Okay, meron tayong DNA sa katawan. Sa each cell, madaming mga DNA. And then pag nag-replicate siya, kukuha siya from the parental strand. And then gagawa siya ng new template hanggang sa mag-replicate yan. Okay po. The semi-conservative model, in the daughter cell, one strand is derived from the mother while the other strand is newly synthesized. Each strand serves as a template or mold. So imagine niyo nasa factory, meron kang mold na panggawa ng bola, for example. Tapos doon sa mold na yun, gagawa ka ng another product mo na bola. Okay, parang may mold. Doon siya mamumold. Okay? Over which a new complementary strand is synthesized. Doon po tayo gagawa ng panibagong DNA doon sa mold na nanggaling doon sa parental DNA. Okay po? Remember that replication always happens from 5' to 3' direction. I cannot stress this enough. Pakitandaan po, this will be applicable sa ating mga susunod na principles. Okay? Kaya po, dito pa lang, from the very beginning, pinapaalala na natin na replication always happens from 5' to 3' direction. Kasi, applicable nga yan sa mga susunod na principles natin. Okay? You will find out later on. So, ano ba yung sinasabi natin na nanggagaling siya sa parental strand? Okay? For example, meron kang DNA dito. And then, gusto na mag-replicate ni DNA mo. So, meron kang 5' and 3' and so, anti-parallel sila. Dapat 3' and dito and then 5' and dito. So, kapag gagawa na ng panibagong DNA ang inyong old na DNA, itong mga blue galing yan dito. So, ito si old number 1, old number 2. Okay? E di nag-replicate na, nakagawa na ng old number 1. Ito naman si young number 1. Okay? Ito naman, si old number 2, ito pala si old number 2, si blue. And then ito naman, young number 2. Gets po ba? Okay? Ang ibig sabihin ni semi-conservative model, doon sa luma mong DNA, nakagawa ka ng panibagong DNA, but it still consists of the old plus the new. Okay? May old ka, may new ka. Doon sa panibago, may old ka, may new ka. Yun po yung semi-conservative model. And then the replication is bidirectional and semi-conservative. We cannot stress this enough. Bidirectional and semi-conservative. Anong ibig sabihin ng bidirectional, ma'am? Bidirectional means that replication begins at a site of origin and simultaneously moves in both directions from this point. Okay, so kay prokaryotes daw po, one site lang yung origin ng each chromosome. Pag eukaryotes, katulad nating mga humans, meron tayong multiple sites of origin on each chromosome. So, pakitignan po yung picture natin dito. For example, meron kang DNA dyan. Kung magre-replicate yan, may mangyayaring replication sa DNA na yan, bidirectional, ibig sabihin magre-replicate siya papunta dito, kasi parang magbubukas yan eh. Magkakaroon ka ng replication bubble, magkakaroon ng fork. Magbubukas din yan papunta dito and then papunta sa kabilang side. Bidirectional, ibig sabihin dalawang side mag-replicate dito, may mangyayaring replication dito. Okay, ganun po yun. And then, ang ibig sabihin nitong eukaryotes, may multiple sites. Imagine niyo sa loob ng katawan natin, may mga cells, doon sa nucleus, may DNA. Isipin niyo yung DNA nyo, nagre-replicate siya sa iba't ibang sites. Sa prokaryotes, katulad ng ating mga bacteria, meron lang silang site. May specific site sila kahit sobrang dami pa nilang DNA. Meron lang silang specific site kung saan sila mag-uumpisa ng replication. Pero tayong mga eukaryotes, maraming site. So kung dito sa site na to, pwedeng mag-umpisa na yung replication, doon naman sa kabilang site, pwede na din mag-start yung replication. Walang particular, walang sinabing isa lang. Multiple sites nga po. Okay? Ang tawag po natin dito, origin of replication. Wala namang criteria to choose kung saan mag-uumpisa. Basta doon. Doon lang siya mag-start ng origin of replication. Pero, walang criteria, pero meron isang basis on where to start. Doon po tayo pupunta sa isang part ng DNA kung nasaan yung meron tayong adenine and thymine rich na site or nitrogenous base. Okay? Hindi tayo pupunta doon sa ating, hindi magsa-start ang replication kung nasaan si cytosine and guanine. Bakit po? Feeling nyo, bakit dapat siya mag-start sa adenine and thymine rich area? And the answer is tama. Tama yung nasa isip mo. Kasi meron ka lang 2 hydrogen bonds sa adenine and sa thymine. Pag ibig sabihin, pag dalawa lang yung hydrogen bonds mo, it will require less energy para mag-start yung replication. Di katulad kay cytosine and then kay guanine, tatlo yung hydrogen bond, mas matagal ka pang, mas matagal mo pang ma-open, mas matagal pa mag-start yung replication bubble, yung fork, and then mag-start sa logging, di ba? Compared dun sa adenine and thymine, 2 hydrogen bonds lang meron siya. So, mas madaling makapag-start yung replication. Okay? Wala naman pong basis, like what I've mentioned, walang sinabing particular place kung saan siya dapat mag-start. Ang importante lang, isang basis lang is dapat nandun ka sa adenine and thymine rich area. Doon mag-start ang ating replication. Okay po. Next, for the prokaryotic DNA replication, meron tayong video dito na papanoodin. So, ano lang natin. Puntahan lang natin siya. And let's all watch this one.
Like these prokaryotic E. coli cells, replicate themselves quickly and efficiently. Part of the process of asexual reproduction is the ability of cells to make identical copies of their DNA before cell division occurs. Prokaryotic cells that reproduce by binary fission rely on the fast, accurate process of DNA replication to ensure future generations of cells will have the same genetic instructions as the parent cell. The structure of DNA aids in the speed and accuracy of replication. Double-stranded DNA is a polymer of 2 strands of nucleotides, which are hydrogen bonded to each other to form a double helix. Nucleotides are molecules that consist of a deoxyribose sugar, a phosphate, and one of 4 nitrogenous bases. The phosphodiester backbones consist of alternating sugar and phosphate groups. The nitrogenous bases include cytosine, thymine, adenine, and guanine. Cytosine forms 3 hydrogen bonds with guanine. and thymine forms 2 hydrogen bonds with adenine. This is referred to as complementary base pairing. The double helix will have one strand oriented in a 5' to 3' direction relative to the hydroxyl group of the deoxyribose sugar, and the other strand oriented in a 3' to 5' direction. This shows the anti-parallel nature of the DNA strands. The complementary base pairing in the structure of DNA allows replication to be executed in a semi-conservative manner. Each strand of the DNA molecule is used as a template in the creation of a new double strand. Replication begins with double-stranded DNA being separated, and each original strand, called a parent strand, is used as a template for the complementary base pairing of nucleotides to make 2 new molecules. DNA replication occurs in the 5' to 3' direction, adding new nucleotides to the 3' end of the newly forming strand. DNA replication will begin at a specific area of the molecule called the origin of replication. The origin of replication denotes the area of active replication called the replication fork. In order to understand how complex eukaryotic organisms replicate DNA, scientists first studied replication in prokaryotic models like E. coli. A number of enzymes are needed for replication to proceed once the replication fork is established. Helicase separates the strands of the double helix, and single-stranded binding proteins stabilize the newly single-stranded regions. DNA gyrase is used to make sure the double-stranded areas outside of the replication fork do not supercoil. Once the replication fork is stable, DNA polymerase catalyzes the addition of new nucleotides to the growing daughter strand. Other proteins, such as beta clamps and the clamp loader, help hold the DNA polymerase in place on the DNA. Short sequences of RNA, called primers, have to be paired to the template strands by the enzyme primase because DNA polymerase cannot begin to add nucleotides without a primer. Replication of both strands occurs at the same time, one using continuous synthesis and the other discontinuous. Continuous synthesis occurs on the 3' to 5' oriented parent strand, referred to as the leading strand. New nucleotides are added to the 3' end, moving continuously toward the expanding replication fork. Discontinuous synthesis occurs on the parent strand that is oriented 5' to 3', called the lagging strand, and is completed in segments called Okazaki fragments. Replication on this strand uses primase to add primers ahead of the 5' end of the lagging strand. DNA polymerase III then adds short sequences of nucleotides, the Okazaki fragments, to the primer, filling in the gap. As the helix is opened further, this process repeats until the entire strand is replicated. DNA polymerase I replaces the RNA primers with DNA nucleotides, and DNA ligase is used to ensure bonding between the fragments and the replaced nucleotides. Once both the leading and lagging strands have completed their replication, 2 identical copies of the DNA molecule result. The process of DNA replication allows actively dividing bacterial cells to make sure all daughter cells have the same genetic instructions as the parent cell, allowing them to function in the same manner. Thus, bacterial populations can grow, increasing the number of individuals in a colony.
So we were able to witness the prokaryotic DNA replication. It has its similarities when it comes to the replication, DNA replication of the eukaryotic cells. Meron lang silang difference sa enzymes or may mga steps lang na hindi applicable to eukaryotic cells. But basically, they're the same. And ang main difference lang nila is the replication of the prokaryotic cell happens in the with the binary fission. Wow. Meanwhile, for the eukaryotic cells, ang tawag naman doon sa atin ay mitosis. Okay, so balikan lang po natin. May nakalimutan lang akong sabihin. So for the replication, for the DNA replication of the eukaryotic cells, usually saan ba nagsistart yon? Di ba sa chromosome? Sa S phase. S phase. Okay, S phase po yung tawag kung saan nagsistart na yung DNA replication. Sa ang particular stage na ba yan ng ating mitosis? Okay, ano pong tawag doon kung nasaan ang inyong S phase? Pakiisip po, saan po nagsistart? Okay, so particularly from the G1 and then S phase, dandun po ang DNA replication. And then next, yung mitosis, and then it will be divided into cells na. So magiging two daughter cells na siya from the mitosis. So particularly sa S phase, okay, doon po nangyayari ang ating DNA replication. So again, for the prokaryotic cells, one site lang ang kanilang origin of replication, and then binary fission ang kanilang pagde-divide. Sa eukaryotes naman, ang tinatawag sa atin ay mitosis. And then we have multiple sites where we can begin our DNA replication or what we call the origin of replication. Particularly kung saan merong adenine and thymine rich area of part of the cell. Kasi nga po doon may two hydrogen bonds and it will require less energy para makapag-start ang DNA replication. For the prokaryotic DNA replication, binary fission, and then you have your DNA gyrase. Okay, the DNA gyrase or gyrase is a type 2 topoisomerase in the prokaryote. So katumbas sa eukaryotic cells nito ay ang tinatawag nating topoisomerase. Okay, and then it may introduce negative supercoils in resting circular DNA, or the negative supercoils in eukaryotes result primarily from packaging DNA into the nucleosomes. And then it facilitates future replication. The negative supercoils neutralize the positive supercoils introduced during double helix opening. So basically, our replication, it will be summarized into 3 steps. Madami mo po siyang dadaan ng steps later on, but basically, ito lang yung summarization of the DNA replication. It will start from the initiation, the elongation, and then the termination. Again, initiation, elongation, and the termination. For the eukaryotic cells, it replicates 100 base pairs per second. And then for the prokaryotic cell, we have 1,000 base pairs per second. So imagine, mas mabilis mag-replicate ang ating mga prokaryotic cells, your bacteria. So mas mabilis po silang mag-spread sa ating katawan and then mag-cause ng particular disease. And then for the eukaryotic DNA replication, the replication fork formation, it is the unwinding or the separation of two strands to form a V. Okay, kung natatandaan doon sa ating prokaryotic DNA replication, nagbuka yung DNA, 'di ba? Nag-form ng V where active synthesis occurs or what we call the replication fork. So if you're having a hard time to imagine ano yung sinasabi ni Ma'am na nagbuka, meron po tayong mga animation later on na ipapakita. Again, for the DNA replication of our eukaryotic cells, it will always be bidirectional and semi-conservative. Meaning, kahit na madami tayong multiple sites of origin of replication, parati po siyang bidirectional, meaning it will go on the right side and on the left side. Pero ma'am, bakit po ganun? Kanina ba po nire-remind na dapat po ang replication, it will start from the 5' end and then until the 3' end. Eh bakit po dito sa ating DNA strand, di ba po dalawa po sila, anti-parallel po sila. Yung isa po doon may tendency na magkaroon ng 3' end, yung una 5' end yung dulo. Paano po mag-take place ang ating DNA replication kapag ganun? Okay, diyan na po papasok ang ating mga enzymes and other steps that will aid our DNA replication. May mga enzymes na po natutulong para mag-take place pa din ang ating DNA replication despite the difference between the 3' end and the 5' end. Okay, so kahit hindi po sila 5' end and 3' end, meron po tayong mga enzymes na tutulong. Kasi dito walang problema. 5' end to 3' end, okay po. Pero pag 3' end and then to 5' end, medyo may konting problema na. Pero with the help of our enzymes, mapapadali ang buhay ng ating DNA replication. So let us just watch this animation video kung saan mag-start tayo sa ating DNA replication. So kung nakita nyo, napanood nyo dun sa ating prokaryotic DNA replication, nagbumuka. Bumuka yung ating DNA, di ba? Parang nagkaroon ng replication bubble and then meron kang old or template na DNA and then nagkaroon ng panibagong DNA strand. So, ganito din. Same din siya sa eukaryotic cells. Okay, let us just watch. It will start from the replication origin. Meron po tayong tinatawag na pre-replication protein complex. Doon talaga magsa-start ang ating origin of replication. So, pre-replication protein complex or PRPC, it will bind and separate the DNA. Doon siya nagsa-start. Dapat doon tayo sa adenine and thymine rich area and then magkakaroon na ng lagging yan. Magkakaroon na yan later on. Yung pre-replication protein complex, wag pong kalilimutan, it will bind and separate the DNA. So, nung nabumuka na, nung nagkaroon na ng parang mga proteins doon, pre-replication protein complex, bumuka na, nagkaroon na ng replication bubble yung mga fork dito. Okay, itsura po siyang fork. And then next, let us just watch this animation video. So, meron na tayong pre-replication protein complex doon sa ating origin of replication. Huwag niyo pong pansinin yung helicase, hindi po helicase ang tawag dyan. And then, meron tayong mga tinatawag na single-strand binding protein. Okay, meron sila mga purpose. Ang ginagawa nila, it binds to the parental strand, number 1. Number 2, it prevents parental strand from re-annealing or reconnecting. Kasi may mga tendency talaga na magdidikit yung mga parental strand eh. Parang may tinatawag bang magnetic force. Hindi naman siya magnetic, pero parang may magnetic force. So, ang purpose ni inyong SSBP, mga single-stranded binding protein natin dyan, pinipigilan nila yung mga parental strand na magdikit. Okay? Kasi gusto nila magdikit eh. So, ang purpose nila dyan, it will bind to the parental strand. It will prevent the parental strand for re-annealing or reconnecting with one another. And then, it will act as a barrier to protect the parental strand from exonucleases. So, may mga lalapit po kasi dyan na exonucleases na tinatawag natin. So, yun yung trabaho ni SSBP, single-stranded binding protein. Tatlo po yung kanilang main purpose. Number 1, ulitin lang natin, that is to bind to the parental strand. Kitang-kita naman, nakabind sila dyan. And then it will prevent the parental strand for re-annealing or reconnecting. Kasi nga ang purpose is paghiwalayin, makagawa ng parental strand para makagawa ng template for another or daughter DNA, di ba? And then it will act as a barrier to protect the parental strand from exonucleases. So once na meron na tayong replication bubble, imagine natin may mga single-stranded binding protein tayo dito. Okay, may replication bubble na tayo dyan. Nakabuka na. Meron ka ng fork dyan. Next step natin dito, take down notes po and then mag-focus po tayo. Medyo complicated na tong second part ng ating lecture video. Nandito na yung pinaka-complicated part of the DNA replication. So makinig po na maigi sa ating second part lecture video.